Ladies and gentlemen, welcome to today's webinar on multiple reaction monitoring using SpectroDive. My name is Florian Marty and I'm your host today. With me joining us for the question and answer series at the end of this webinar will be Lucas Reiter, Chief Technology Officer of Biognosys. As always, in our webinars, he will be available for questions afterwards and discuss the topics. Today, I will give you first a brief introduction about next-generation proteomics with a focus on multiple reaction monitoring. We will cover the basic principles for relative and absolute quantitative MRM and provide you with some applications that are published based on Biognosys products by our customers. Then, we will have a closer look at the Biognosys products for multiple reaction monitor with a focus on our proprietary software SpectroDive. Further, we will look at an in-house conducted study where we did absolute quantification of plasma proteins using Biognosys quantification kits, PlasmaDive and Plasma Deep Dive. And last but not least, we will have time to discuss some of your questions. Like any other webinar presented by Biognosys, this webinar will be recorded and sent to all participants afterwards. If you want to know more about the workflows offered by Biognosys or have questions that were not addressed in the webinar, please contact us at support@biognosys.ch. The proteomics challenge is defined by the challenge bridging the gap between scale and accuracy in protein analysis. It simply means the current workflows need to be further developed to overcome these limitations. Nowadays, we have on one hand methods that are able to identify a lot of proteins in one single measurement, but have a very poor ability to quantify. On the other hand, we have antibody-based ELISAs, which are very good for quantifying certain amount of proteins in low throughput. With the Biognosys next-generation proteomics technologies, HRM and targeted MRM, we are trying to bridge this gap and enable our customers methods that allow them to quantify a high number of proteins with very reproducibly and accurately in thousands of samples with high throughput. For these two workflows, we have a number of products in-house. On the one hand, for the HRM-MS, the discovery-based workflows, we have sample preparation kits, then the IRT kit for retention time normalization, and the proprietary software SpectroNow to do data analysis on the DIA-based datasets. On the other hand, multiple reaction monitoring, or MRM, is the targeted proteomics workflow that wants to elucidate the question, what happens to my protein of interest? It takes advantage of SpectroDive to generate MRM methods that are used to acquire data on any mass spectrometer capable of doing MRM or PRM. For retention time normalization and calibration, it is recommended to also use the IRT peptides in these kind of workflows provided by Biognosys. After successful data acquisition, SpectroDive is used to perform data analysis and determines either relative or absolute quantities of the candidate proteins. In a label-free relative quantitative MRM assay, predefined peptides of a given protein are specifically selected in Q1 and transferred to Q2, the second quadrupole where fragmentation takes place. Out of these fragments, a pre-selected number are further analyzed in the third quadrupole and the spectra is generated. By selecting multiple proteins and fragmenting the respective prototypic peptides, relative quantitative information of the selected proteins can be acquired. The power of this workflow has recently been demonstrated by Alessandro Ori and colleagues. In a multi-omics approach, they were interested in protein deterioration in old rats versus young rats. Among the technologies used, they have used relative quantitative MRM to establish the proteomic levels of certain proteins. In a similar fashion as relative quantification, one can also obtain absolute quantitative information using multiple reaction monitoring. To do so, selected reporter peptides of a protein of interest are chemically synthesized with heavy labeled lysines or arginine residues. These stable isotope labeled peptides are then spiked into the sample at known amounts. In the next step, both the light and the heavy labeled peptides are analyzed with high precision and selectivity using multiple reaction monitoring. In SpectroDive, the data is then analyzed and the ratio light to heavy is determined to calculate the absolute amount of your protein of interest. As you will see later in this presentation, in SpectroDive, this is done on the fly during the data analysis. Von Appen and his co-workers have used this approach in combination with cryo-electron microscopy to elucidate the composition of the nuclear pore complex. This elegant study has been recently published in Nature and is a good example to show the power of absolute quantitative information that can be gained using Biognosys Products and MRM. In order to enable our customers the same proteomics tools as used by Ori and Von Appen, we have developed specific products. For relative quantitative approaches, we developed SpectroDive to do the method generation and data analysis. Together with the IRT peptides for retention time normalization, you have a complete workflow that enables you to acquire relative quantitative information with MRM. On top of that, we also have developed absolute quantitative proteomics kits that enable you to do absolute quantitative research on human plasma. We will further have a look into these later. The most important tool for doing quantitative MRM experiments is a dedicated software. At Biognosys, we have developed SpectroDive, the easiest and most powerful tool for targeted proteomics. It allows you in a quick and easy manner to set up MRM and PRM measurements. It has the smartest peak picking algorithm out there for fast data analysis with minimal bioinformatics resources. With the benefits of the IRT peptides, you get higher data confidence with complete workflow quality controls. And using stable isotope standards, you can get integrated absolute protein quantification. In the next couple of slides, I will walk you through SpectroDive and show you how SpectroDive is built up to do the MRM method generation, data analysis, and quality control. SpectroDive is designed in a way that it follows the logical analysis flow of an MRM or PRM experiment. In the first perspective, the so-called prepare perspective, the researcher can generate the MRM or PRM method. On the left hand here, the researcher imports the MRM panel, which is used either for PRM or MRM on the mass spectrometer of choice. Down here on the third window, one can then adjust the method and export the method that can be easily read by the mass spectrometer, and the MRM data can be acquired. In the next perspective, the so-called review perspective, the researcher can look into the data and see whether whether peak picking and everything else worked out fine. The researcher here has a full flexibility to change all the information provided by the software. He can decrease or increase the boundaries which were used for peak picking and accept or decline transitions that were used for quantification. Further, IRT refinement done in the software improves the quantitative accuracy. On top of that, in experiments where stable isotope labeled peptides were spiked in prior, the software is able to determine the absolute quantitative amounts of your protein of interest. Unless other MRM analysis softwares, SpectroDive also contains a post-analysis perspective. In this perspective, the differentially abundant candidates are listed and plotted in a volcano plot. In the next perspective, the report perspective, all the data can further fully flexibly be exported and used for any kind of downstream analysis. On top of that, SpectroDive also contains a so-called quality control perspective where the performance of the LC-MS is monitored over time using the IRT peptides. In the next couple of slides, I would like to show you now how we at Biognosys use our softwares and kits to perform absolute quantitative proteomics experiments. We have conducted a study to determine the absolute concentration of plasma proteins in healthy subjects. For this, volunteers of both genders were chosen and samples were collected once a month over a time of 4 months. Together with the sample collection, the participants had to fill out a questionnaire about general health status. All samples were then processed and measured in-house according to our pipeline. In a first step, we have done a shotgun proteomics approach together with literature research to generate a catalog of potential plasma biomarkers. Prototypic peptides were then selected and stable isotope labeled standards of these peptides were synthesized and ordered. Further, MRM assays were refined to lead to an MRM panel called PlasmaDive. The PlasmaDive panel quantifies 100 human plasma proteins from serum or plasma. It can be used with a sample volume as little as 10 microliter with or without depletion. It contains one stable isotope per protein and quantification is done on the peak area of 3 transitions over several orders of magnitudes. With or without depletion, this leads to high accuracy of your quantitative information in absolute manner. To further increase the quantitative depth of proteins measured in plasma, a second panel was developed. Together, the PlasmaDive and the Plasma Deep Dive panel span 7 orders of magnitudes for 175 proteins. These panels were then used to measure the 9 healthy donors explained before. As indicated here, the panel contains proteins that are stably expressed and have very low CVs between individuals, and thus resemble housekeeping genes used in genomic applications. Moreover, there are also proteins that show differential regulation either between subjects or proteins that can be used, for example, as pregnancy biomarkers. In this donor, we see that one of the proteins goes up during the pregnancy and is therefore used as a marker. In summary, the study conducted in-house demonstrates the power of multiple reaction monitoring for the validation of target proteins and biomarkers. The amount of information that can be obtained with the next generation proteomics workflows provided by Biognosys can further be extended. Using hyperreaction monitoring with SpectroNaut, it is possible to use heavy labeled panels such as PlasmaDive in combination with label-free discovery proteomics. This allows the researcher to gain absolute quantitative information of predefined proteins and relative quantitative information in a discovery proteomics experiment. Moreover, the absolute quantitative information of the spiked-in peptides can be extrapolated to a certain extent, and the absolute quantitative values can be determined for the entire sample. In summary, I showed you that MRM allows you to relative and absolute quantify proteins over at least 7 orders of magnitude in plasma and serum. Biognosys provides you with products that support the entire MRM workflow. SpectroDive, the software to perform the method generation and data analysis. Further quantification kits such as PlasmaDive, Plasma Deep Dive and PlasmaDive Mini for absolute quantification of plasma proteins. To this, the IRT kit is added for retention time calibration and normalization and can be used within SpectroDive for quality control assessments. The entire MRM workflow can further be extended to hundreds of proteins using the PRM workflow also supported by SpectroDive. Last but not least, a combination of heavy panels in HRM is also possible within SpectroNaut and Biognosys Products. With this, we are coming to the end of our presentation from today, and Lucas and me will now be taking your questions. Okay, the first question by Christine Vogel. Are there other approaches, workflows such as HRM that can cover similar dynamic ranges?
We've never looked at this so systematically. We've never directly compared HRM with MRM, but there are a lot of high-quality publications about this topic. I think at the moment, no technology will probably beat MRM or PRM in terms of dynamic range, but the other technologies are catching up in general, I would say.
Next question. Could you comment on MRM versus DIA in terms of experimental benefits and challenges of both techniques?
Okay, in terms of experimental benefits, HRM definitely shows you the bird eye view, or DIA. So you can use it for discovery experiments, but you can also combine it. You can also do hybrid targeted and discovery workflows. So you can even spike in stabilized substandards, perform HRM or DIA, and combine discovery with an accurate quantification of a number of proteins using stabilized substandards. But when it comes to really quantifying a few analytes with very high throughput, very high dynamic range, and also very, very high precision, even on an absolute scale or even for absolute quantification, then there is still no way around MRM and PRM. But as I said, there's not so many drawbacks anymore for HRM in terms of performance. It's combinable with targeted approaches. You can have hybrid approaches, and yeah, in terms of setting up the experiment, MRM and PRM is more complicated because you have to have better assays, and typically also more manual effort is put into those assays, especially if you want to order stabilized substandards, because then it gets expensive and you want to make sure that you select the right peptides for that.
The next question, can you please tell us more about your kit for plasma and absolute quantitation? As I mentioned in the presentation, the SpectroDive spans 100 proteins over certain over 4 orders of magnitudes. In addition, with the plasma deep dive, 7 orders of magnitude can be analyzed. If you wish to get more detailed information on the proteins that are in the kit or any other information about the kit, please send us your question at support@biognosys.ch and we can give you the answers. Nikhil Pandya wants to know if we had the chance to compare a different number of peptides per protein to verify if our approach of taking one peptide in PlasmaDive gives accurate protein quantification.
We have also not systematically performed that. Also here from literature it is known that the absolute quantitative values you can get if you do that can vary in the range of 2- to 3-fold. Depending on which peptide you select, and it is generally known that a number of factors can have an influence, such as the tryptic digest or modifications on the peptides or peptide degradation and so on and so forth. In general, we see this workflow more as a very precise and high throughput tool to do very accurate relative quantification, and the difference to HRM or DIA here is that you can do this over a very long period of time. So the platform can change completely in between. You can have a completely different LC-MS platform. The data you generate is still comparable across these runs. That's a major difference to HRM or DIA, where it is much more difficult to compare datasets that are 5 years or let's say 3 years old to datasets that are generated nowadays. So that's definitely a major difference. Yeah, and in terms of absolute quantification, if you really want to do absolute quantification of proteins, there is no way around having a protein standard. And that's a completely different league, because having protein standards over a long period of time, that's a very big effort. And that's also not what most people need. Most people need accurate relative quantification over a long period of time and many different samples, maybe even platforms. And that's, if you, in terms of absolute quantification, are roughly in the right range, that's enough.
Along this question, the next question, what criteria were the proteins from PlasmaDive selected?
We want to have a set of interesting proteins that are detectable on any standard system. Yeah, that was the main objective.
Are there any other questions considering the introductionary webinar on multiple reaction monitoring? If not, I would like to thank you all for taking your time to being with us today, and I wish you a nice day.
Thanks, everybody.